hear me? Okay, there we go. So today we're going to take a look at a demonstration on diffusion and osmosis. We're going to take a look at the cells of the other tissue types, the muscles and connective tissue and nervous tissue. We're going to take a look at the skin. And if we have time, we'll go ahead and take a look at the cheek cells. If not, we'll just push that back a week and do it next week. So we have some flexibility. Okay, I hope everybody can hear me. If you can't hear me, send me a chat message. I'm going to go ahead and bring us into the, everybody should see a slide that says membrane transport and permeability. And if you're following along in class here, make sure it says AP1 Lab Unit 1, Week 2, membrane transport and permeability. And I have cut this down significantly. So if you have slides that I'm not talking about, don't worry about it. I've cut it down a lot because yesterday it just seemed like it went on and on and on and on. And even I bored myself. So, oh, wait, I wasn't supposed to say that. Okay, so, okay. Membrane transport and permeability. In lecture today, we were talking about solutions, solutes, and solvents, and things like that. Our cells are constantly being exposed to water. Our cells are 70% water to begin with. Yes.
I may be wrong, but I don't believe you finished before.
Oh, I know. I'm going to work on it today. It's okay. Don't worry about it. So, yeah, I want to finish week 1 today. You know, I always have these great expectations of what I'm going to get done in the first week, and I never get there. So I'm just going to push along as best we can. So thank you for pointing that out. So I'll get you later. So, I'm kidding. So okay. This day and age, you got to be careful what you say, you know. So I know. Okay. So our cells are filled with water. They're 70% water. Our bodies are 70% water. Our blood vessels are filled with plasma that carry blood cells. Plasma is 70% water. It's 0.9% sodium chloride. I mentioned in lecture today about the percentages are very simple ways to measure solutions. A 0.9% sodium chloride solution would be about 9 grams of salt in 1,000 milliliters of water. So it's not very strong, but it's still a percentage of solutions. Now, our plasma is 0.9%. The cells are 0.9%. The blood vessels leak. The blood vessels are not perfect hoses or perfect tubes. They've got lots of holes in them. And so that plasma is always leaking out into the surrounding tissues. That's what they do, because that plasma is going to carry salt and glucose and hormones and anything else, you know, cells that we want to get out to the other cells, like white cells might leak out in the plasma too. So we're always leaking plasma. When it leaves the blood, it becomes what's known as interstitial fluid. It becomes what's known as interstitial fluid. Interstitial means the spaces in between. So it's between the blood and between the cells. So it surrounds the cells. The cells are being bathed by this interstitial fluid. Now, we don't waste that fluid, we collect it. After it soaks all the outside of all the cells, it doesn't go in. It soaks the outside of all the cells. Then it's collected by a secondary circulatory system we have called the lymphatic system. And that collects all the interstitial fluid, runs it through our lymph nodes. You've heard of our lymph nodes, right? Like here and under the arm and, you know, in the chest area and in the groin. We run it through our lymph nodes, we filter it through our lymph nodes. Our white cells hang out in our lymph nodes looking for something to kill. And that's what they do. They either produce antibodies or they kill things. They're looking for pathogens to kill. And then after it's been filtered and cleaned, it enters our blood supply again as plasma. Same water, it was plasma, it was interstitial fluid, it was lymphatic fluid, and now it's plasma again. It just starts over and over again. Now, when it's exposing the cells, we can move stuff back and forth between the cells. And cells don't like to spend any energy. They like for stuff to move freely. And there's two ways that cells move things from water into or out of the cells. One approach is something called diffusion. Diffusion. It's okay, so passive equal free. Diffusion is how we get stuff from the plasma into the interstitial fluid where it can go into the cells, or where something in the cells can go into the interstitial fluid. Diffusion is simply the movement of a substance from a high concentration to a low concentration. I hope I don't run out of word. Low concentration. That's all it is. Passive. It doesn't require any energy for us to move stuff from a high concentration to a low concentration. If I were to accidentally dump over a bottle of a very aromatic chemical, for example, there's an artificial banana flavor that's often used to test respirator packs in the fire department and in rescue squads. They put the mask on, and then someone opens up some of this what we call it artificial banana oil, and if they can't smell it, it means that their mask is working. Essentially, it doesn't get through the mask. So if I were to dump some banana oil over here, and I picked banana oil because it smells pretty good. I could have picked something that smells like rancid goat butter, but banana oil is better. Who doesn't like bananas? So anyway, eventually it would spread out throughout the classroom. It would reach equilibrium where it was evenly distributed. Now, how did that odor get all the way around the room? When I wasn't over there going, come on, you know, it was a passive movement. The air molecules in the air are just pushing the molecules of the chemical around. And it spreads out until it reaches equilibrium. The same thing happens in water. If I add a drop of dye to a beaker of water, eventually the random movement of the water molecules will distribute the dye. That's diffusion. The odors in the air is diffusion. It's all passive, free process, and cells love free. So when we have interstitial fluid surrounding our cells, we want to send stuff into the cells, or we want the cells to send stuff out, and we don't want to spend any energy to do it because cells are cheap. Cells are they're just frugal. They only spend energy on things they need to spend energy on. So diffusion is the process we use to get stuff into or out of the cells most of the time. So this is diffusion, passively through the cell membrane. See it on the far side here in the screen. On the left-hand side, you know, substances can move through the cell membrane. Unfortunately, our cell membrane is made up of fats, it's lipids. So some things have a problem getting through the cell membrane. Other things can go through very easily. If we have a problem, we have to have what's known as facilitated diffusion, where we have a special channel just for a substance and nothing else gets through. And then the third category of movement across the membrane is active transport, where we actually have to spend energy. And we don't like to do that. We like to have things passively. But all we're talking about today is passive movement. We're talking about diffusion. I already said this already. There we go. Diffusion and osmosis. Diffusion, the movement of a substance down its concentration gradient from a high concentration to a low concentration. If I put a pellet of dye in a beaker of water, it would gradually spread out until we were at equilibrium, just like the odor. So didn't take a thing to do that. Now, where do we use diffusion in our bodies? Well, breathing is one. When we suck in air, that air is 80% nitrogen and 20% oxygen. When that air reaches the structures in the lungs where we have gas exchange taking place and blood vessels are right next to those structures, they're called alveoli, and a blood vessel is right next to that, and they're one cell layer thick apart. The oxygen concentration in the air in the lungs is 20%. The oxygen concentration in the blood vessel is about 16%. So we have a higher concentration in the lungs than we do in the blood supply, and oxygen diffuses across the cell membrane into the lungs. And so we recharge our blood supply. At the same time, carbon dioxide is always higher in the blood than it is inside the lungs, inside our air. Because carbon dioxide levels in the atmospheric air here are very, very low, but they're higher in the blood because we've been churning it out of our cells. So that carbon dioxide diffuses across that membrane back into our lungs. And we don't have to do a thing. All we got to do is suck in air and blow out air. All the hard work is done at the level of the cell with the capillary as a free ride. So we're moving air in and out, but the actual exchange of gases is through simple diffusion across a membrane. So we can move gases that way. Nutrients in our small intestine, when we eat stuff and it gets processed and it slides into our small intestine where we're going to start absorbing nutrients. Nutrients move across the membrane of the small intestine into our blood supply through diffusion, because there's always more carbohydrates and proteins and fats inside the small intestine than there is in the blood supply. So it all slides into our blood passively. So we do a lot of things through simple diffusion in our bodies. So simple diffusion, simply moving down the concentration gradient. But the other mechanism we use, you're going to hear more about this in lecture. So I don't want to beat this into the ground. There's another term called osmosis. Osmosis is a specialized type of diffusion. It's still free. Osmosis is the movement of water across membrane, a cell membrane. From high water to low water. Now, what do I mean by high water and low water? Well, I've got an example set up here, a little experiment running. I'll show you here as we go through the next hour or so. I've made up a 10% salt solution, 10% sodium chloride in one beaker, and the other beaker is filled with distilled water. Now, a 10% salt solution is also 10% salt and 90% water. That equals 100%. Okay, I think I checked the math on that. So there is a solution, 90% water, 10% salt. Now, distilled water, do it over here. The distilled water equals 100% water. If I have a membrane, this is a membrane. It's a highly technical drawing of a membrane too, by the way, okay, just to let you know. If I have 100% water here, and I have 10% salt and 90% water here, which side of the membrane has the higher water concentration? The 100%, right? 100% water. This is 90% water. So water will automatically cross the membrane and go this way. In osmosis, water always moves from the higher water concentration to the lower water concentration, regardless of what we're dealing with. This could be, I could have another membrane here where I have 60% water, 40% glucose. And over here, I could have 30% water and 70% salt. Even though 60% water is not very much, it's still greater than the 30% water. Whatever side of the membrane has the higher water concentration, water is going to go from the high side to the low side. That's a little factoid. Always remember that. So if the water concentration inside our cell, for example, in a human cell, the salt concentration in our plasma is 0.9% sodium chloride, which means that it's 99.1% water. 99.1% water. If the water concentration is greater than that, water is going to go into the cell. If it's less than that, it's going to come out of the cell. Let's say that you had a patient who has been vomiting extensively for 24 hours. They become dehydrated. They're losing water from their plasma because when they're vomiting and they have diarrhea, the water loss is coming from their plasma. So you're becoming dehydrated. You've lost water. Now our bodies don't lose salt. Our bodies, the actual amount of salt present in our body stays fairly constant, but the concentration changes because we gain or lose water. So if you've got a patient that's been vomiting for 24 hours and they're losing a lot of water, or maybe they've got a high fever and they've been sweating profusely for 24 hours, they're losing a lot of water, but they're retaining the salt. Their salt concentration is rising because the water levels are dropping. So if they're losing water, which way is water going to go? Into the cell or out of the cell? Water always goes from the higher water side to the lower water side. One of the problems we have with dehydration is when we lose water from the plasma, the cells have to give up water to try and make it up. And that damages the cells. Dehydration hits us in a variety of ways, and one of which is that cells start to shrink if they have to give up water. And cells don't like that. So anyway, I have a little demonstration running here for you to show you this. Simple diffusion, we move from the high concentration to low concentration, but in osmosis, we go from high water to low water. Now these two beakers here, the beaker that has a label that says 10% salt, that's a 10% sodium chloride solution. And inside the beaker is an artificial cell. Now this artificial cell is made up of what we call dialysis tubing. Dialysis tubing is a filtering tube with extremely tiny holes in it. Now, water's not, these are from yesterday, but these were filled. One was filled with distilled water and one was filled, this artificial cell was filled with salt water. It doesn't leak out because the holes are very tiny. They're microscopic holes. We use them for filtering in dialysis equipment. We have these tubes here. Here they are. This is what it looks like. Extremely thin. I've got 35 feet of this, and something, anything that's under 3,500 molecular weight will pass through here. Anything bigger than that will get stuck. So things like glucose and salt and water will pass through there, but other molecules larger than that won't. Now, I've put an artificial cell in the distilled water, and inside the artificial cell, the cell contains 10% sodium chloride. Now, if osmosis works, water should go from the high water side into the lower water side. The high water side is 100%. If I put a artificial cell containing 10% sodium chloride in distilled water is 100%. The water concentration in the cell is 90%. Water should go into the cell because 100% is greater than 90%. And what I did was I weighed the artificial cells before we started. The cell with the sodium chloride in it weighed 33.8 grams. If water goes into that cell, we should see a weight increase over the next hour or so. The other beaker was 10% salt water, and I put in a cell that's 100% distilled water. And so we have 100% water in the cell and 90% in the beaker, and that cell should lose weight if it's going to work. So, and if I put the cell in the right beaker, so, which is always a possibility. So, and we'll let it run for a while, and what will happen, and it worked very nicely yesterday, is that the salt water, the cell containing the salt water gained water and gained weight. The cell that contained distilled water and placed in the salt water lost weight because water moved across the membrane. So, and it, it usually works. Sometimes it doesn't, but it usually does. And we've done this in the past where we've had everybody do one, and we've used different molecules. You can use starch, and you can use glucose, and you can use salt. You can mix them up so you have salt inside a beaker and glucose inside the cell. It doesn't matter. Solutes are solutes, whatever the water level is, the highest water level, it's still going to work. So, but we'll check this in about 2 o'clock or thereabouts and see what the weight gain is. It's very simple. If the weight goes up, it's because water was going into the cell. If the weight drops, it's because water was leaving the cell. And that's what osmosis does. I haven't done it. I've done nothing to those beakers. They've been sitting out here all morning. They're just salt water in one and distilled water in the other. So, and a point I always make, and I need to do this one day. If I take this beaker full of salt water, when it was made up, it had 100 grams of sodium chloride in a liter of water. And so that gives us a 10% solution. If I let that sit, well, half the water evaporates. So I went from a liter to 500 mils of water. I still have 100 grams of salt in there. The salt concentration is now 20%, not because I added anything to it, but because half the water left. And if I let it go further, eventually all the water will evaporate and I'll have all the salt left behind. So, and our bodies are like that. We generally don't lose a lot of salt, but our salt concentration goes up and down based on our water levels. Our bodies use salt levels to tell us what our water levels are. When we get thirsty because our salt concentration has gone up. We don't monitor it's hard to monitor our water levels. It's very easy to monitor salt. So, because we run all of our blood through our kidneys 60 times a day, the entire blood supply 60 times a day. And we're always monitoring salt levels and telling the hypothalamus, and the hypothalamus can say, hey, you know, we're a little salty. We probably need some water. We get thirsty. And we bring it back to where we're supposed to be. So anyway, this is, we measure our dialysis bags at the start and at the end. And we have 2 beakers. We have a dialysis bag that's 100% distilled water in a beaker full of 10% salt. And in the other beaker, we have a 10% salt solution in the dialysis bag in a beaker of 100% distilled water. See what happens. It should work. So, questions on this? Okay, we're doing real science today, you know. Okay, let me, now, let's move on and go into the slides. Let me see what's out there. Memory transport. Okay, let me go. See if this comes up. There we go. Okay, can everybody that's zooming see the slide? It says human tissue types, muscle, connective, and nervous. If you can't, let me know. I'm gonna make a bold assumption that everybody can see it and go on. Now, last week, we took a look at the epithelial cells that, you know, we used to, come on. There we go. Last week, we took a look at epithelial cells. They were one of the 4 tissue types. There's only 4 tissue types in our bodies. Epithelial, which are the covering cells. Then we have muscle, connective, and nervous tissue. There are 3 types of muscle cells. We have skeletal muscle. That's voluntary. We control what we do with our skeletal muscle. We can raise our arms, we can lower our arms, we can run, we can talk, we can walk, we can eat. We have voluntary control over those muscles in our arms and our legs and anywhere else that we, you know, tell us to do things. Skeletal muscle is what we call striated muscle. It has a series of light and dark bands, like stripes on there. It's very distinctive. You know, it's the only type of muscle that we have control over. Cardiac muscle is involuntary. We don't have to tell it to do anything. We don't tell the heart to do anything. The heart controls itself. And cardiac muscle is found only in the heart. I guess that's why they call it cardiac muscle, because it's only in the heart. Smooth muscle is found in the digestive system. It's found around all of our blood vessels. It's also involuntary. It controls the movement of food through our digestive system from the time we swallow it to the time we defecate it. It controls the size and diameter of our blood vessels so we can move blood around. You know, we can shrink blood vessels or we can dilate blood vessels. They can enlarge. But see, we don't have enough blood to fill up every vessel in our body. We're always about a unit or 2 short of filling everything up. So we have to move blood around to where we need it and where we want to use it. We don't send them on a cold day, for example. You wouldn't send a lot of blood to your skin because you'd lose a lot of heat. So you restrict the flow to the skin on a cold day. But on a hot day, you send a lot of blood to the skin because you want to get rid of extra body heat. So you're moving it around. When you eat a meal, you know, you're going to send more blood to the digestive system so you can absorb more nutrients. When you exercise, you're going to send more blood to your muscles to meet the demands for glucose and for oxygen and to take away waste products like carbon dioxide. So we're always moving our blood around to meet what our needs are. And smooth muscle lines all of our blood vessels and allows it to open up or to, you know, constrict, not shut completely, but to reduce the flow so we can use blood somewhere else because we just don't have enough to fill up every vessel. Okay, this is skeletal muscle. Skeletal muscle is characterized by a lot of light and dark bands called stripes or striations. So we see, very prominent feature there, all the stripes along in there. Skeletal muscle, and we'll get into this in lecture, skeletal muscle works by sliding over top of itself. And that those light and dark bands are going to slide. And that's why we have that, that's why we have that arrangement. Now, when we look at skeletal muscle, we're going to see a lot of fat cells, because fat is a great place for stored energy. We're going to see a lot of we're going to see multiple nuclei, lots of nucleuses, lots of nuclei present. When we are laying down our muscles, when we are in utero, we take lots of smaller cells and put them together to make our muscles. We keep the nucleus there. We don't get rid of the nucleus. We don't have to worry about the DNA because muscle cells, skeletal muscle cells, don't reproduce. All the muscle cells we had when we were born are what we have now. We didn't make any new ones. Our muscle cells can get larger or they can get smaller. We can exercise and make them big and powerful, or we can not exercise and make them small and weak or weaker. But we don't grow new ones. If the cell is damaged, if a muscle cell is damaged, then we get scarring instead, but we don't make new cells. So here we see skeletal muscle. There are the striations right there. There's some adipose. Our muscles use a large amount of energy all the time. So we, adipose tissue with lipids stored inside are great sources of energy. We get more energy from fat, from lipids, than we do from carbohydrates. Even though we use carbohydrates as our main source of energy, a gram of carbohydrates gives us 4 calories of energy, but a gram of fat gives us 10 calories of energy. So we should, but instead our bodies opt for the carbohydrates instead. Here we see, that's a nucleus. Those are multiple nuclei. Let me get back. Those are multiple nuclei. So because we formed our skeletal muscle cells out of a lot of smaller cells pushed together. So nothing quite looks like skeletal muscle. Now, one of the things about skeletal muscle, though, is that we can control how many cells we're using to do something. We can, we don't use every cell in our arm, for example, in our biceps, this muscle that sits on top of the arm right here. When we do something with our arm, we don't use every muscle cell in that biceps. We only use enough cells to do what we want to do. We can control how many cells we're using. And that's something we don't have in the other muscle cells. With the other muscle cells, it's pretty much all or nothing, particularly in the heart. So we don't have any control over those. Now let's take a look. This is smooth muscle right here. Smooth muscle is different. First of all, no striations. And it's only 1 nucleus per cell. So, and the cells, instead of being striated, are shaped like a spindle, thick in the middle and pointed at each end. And they're lined up, layer on one on top of each other, but they are not, they don't have, each cell has its own nucleus. So like there. Or it looks like this. Again, this is smooth muscle right there. Layer upon layer upon layer. And a close-up of that again. Now, where would we see this smooth muscle? Well, this is a cross-section of the small intestine. And as part of the digestive tract, we have layers of smooth muscle in here that squeeze and squeeze to push food through. From the time we swallow it into the esophagus, to the time it comes out as waste material at 25 feet away, we're going to be squeezing that tube, squeezing and churning, first for digestion and then for absorption and then getting rid of the waste products. And so we see our smooth muscle layer in the small intestine runs in a ring around the tube. Think of the small intestine and large intestine as a tube about 25 feet long, with a layer of muscle that runs in a circular pattern down the tube and one that runs lengthwise. So we can squeeze in two different directions at the same time. And that's what we're seeing here. We are pushing these food items that are from start to finish. We also have the same arrangement around our blood vessels. We want to be able to squeeze our blood vessels to narrow their openings, to restrict flow, or to open them wide to let a lot of blood flow through it to an area. So that's where we see, these are the two big areas where we have our smooth muscle. It's under involuntary control. Our nervous system takes care of this for us. We don't have to think about it. Now, the third category is the heart. We get into AP2, we'll spend a lot of time talking about the heart. Now, in the heart, we have striations. That's the only similarity that the heart has with any other muscle type. It is also striated, but here the cells are very short. They also branch apart. Notice the branchings in here. One cell will lead to two cells, and those two cells will lead to two more cells, and so on. They also only have one nucleus. And where they branch from, they branch from a structure known as an intercalated disc. Now, the thing about cardiac muscle cells is that cardiac muscle cells communicate to each other. The heart, when it contracts, will contract every cell at exactly the same time, at exactly the same strength. We don't pick and choose how many cells we're going to use in the heart, because all four chambers of the heart have to contract together. The two upper chambers and the two lower chambers contract at exactly the same time with exactly the same force. And we're using every muscle cell in the heart to do that. There shouldn't be any stragglers, and there should be no ripple effect as some cells contract stronger than others. They all contract to their fullest extent every time. And even more specifically, the heart has no outside control. The heart is autorhythmic. It sets its own pace. The heart will tell itself to beat every 7/10 of a second for 90 years. The heart will continue to contract and contract and contract. We refer to we describe the heart as acting like one giant muscle cell, as it contracts fully every time. So anyway, characteristic of the human heart: striated short cells, they branch. They look something like this. These little lines here, the intercalated disc, are where the cells branch from. Later on, we'll talk about there are openings between those those lines, so the cells can communicate rapidly to each other. There's cardiac muscle cells, the striations, the intercalated disc, the branching fibers. Fibers and cells mean the same thing in muscles. We call them either muscle fibers or muscle cells. If they branch, branching fibers are the same as branching cells, and we see them branching out like that. That's how they can communicate rapidly to each other. Okay. And again, a single nucleus. But heart muscle cells don't regenerate. They don't reproduce. A heart muscle cell, if it's damaged, it will recover, it will heal up, it will have scar tissue, but it will never regenerate its ability to contract again. A person who has a heart attack and muscle cells die are going to replace those muscle cells with scar tissue, and scar tissue doesn't do anything. It doesn't contract, it doesn't do anything except function as a placeholder, as a patch, if you will. Okay. So those are muscle cells. Now the next category, and we're moving right along here, are connective tissue. Connective tissue. It's three criteria to be considered connective tissue. And connective tissue is one of those things where it's sort of like, we don't know what else where it belongs. Let's make it connective tissue. Because we got things like blood and bone in connective tissue. But the criteria are you have to have living cells in some sort of matrix, whether it's a solid, a liquid, or a gel, with fibers. So you got to have living cells embedded in something or floating in something with fibers involved. And that's what a connective tissue is. So you can have bone cells embedded in a hard matrix, embedded in the bone, and they're alive and they're connected to each other and they have fibers wrapped around them, around the bone to give it strength. You can have blood cells floating in plasma, but the fibers are in solution. They only become active when we're going to form a clot. You can have cartilage. You can have living cartilage cells in a matrix that's like a gel, like soft plastic. It's like cartilage in our knee or cartilage in our nose. We have living cells in there, and we have fibers again for elasticity and strength. So we cover all three criteria, as bizarre as the differences in the matrix can be. Okay. Types of fibers. Collagen fibers. Collagen fibers are the most abundant protein in the human body. There are more collagen fibers than anything else, any other protein we have. Collagen fibers, strand for strand, are stronger than stainless steel. A strand of collagen is stronger than a corresponding strand of stainless steel. Collagen fibers are used to make the tendons that hold muscles to bone, and they are so powerful that they will snap bone before they themselves tear. So we use them for great strength. We wrap our bones with collagen fibers to give them the ability to withstand shock and stress. Elastic fibers, they're made out of elastin. Elastic fibers are stretchable. We find them in places where we want to have a lot of give, like in our skin. Our skin is very flexible. Our skin snaps back into position thanks to the elastic fibers. And then we have what are known as reticular fibers. Reticular fibers form the net that hold all of our organs in place, like here in the abdominal cavity. We have, the liver and the stomach and the small intestine and the large intestine and the urinary bladder and the kidneys. What's holding them in place? How come they don't all sink to the bottom of the abdominal cavity? It's thanks to this mesh framework of reticular fibers holding everything together. So, collagen, mostly used in tendons and ligaments. Muscles pull on bones for us to do things, and muscles attach to the bones by way of tendons. We pull on the collagen fibers before we pull on the bone. The elastic fibers show up all over the place because we have to have a certain amount of give. We have elastic fibers in our arteries. If you've ever taken your own pulse or taken somebody else's pulse or ever had your pulse taken, the pulse is nothing more than the rebounding of an artery in your arm or your leg. When a surge of blood comes out of your heart, it shoots down the arteries. The arteries are elastic, and so the artery opens up as this surge goes through. And as soon as a surge of blood from the heart goes through, it snaps back in place. It goes back to its smaller size. The snapping back of the artery wall is your pulse. It's not the same as your heartbeat. It corresponds to your heartbeat, but it's not the same thing. A heartbeat's something totally different. The heartbeat is the sound of your valves going shut in the heart. But the pulse is, corresponds to the heartbeat, and it's simply the snapping back of the artery wall because the artery wall is elastic. Our vocal cords are elastic. Our vocal cords stretch so that we can alter our voice and make it loud or soft. Or if you're musically inclined, you can sing and hit different notes. We can alter the pitch and the intensity thanks to our vocal cords, the shape of the vocal cords and how they vibrate. Our trachea, our airway, is made of collagen, of elastic fibers. Our trachea is very flexible. Our bronchi, the bronchial tubes in our lungs, are made out of elastic fibers. They're very, very flexible. And then, of course, the reticular fibers, they show up in the squishy organs, like the spleen or the liver. Both have the consistency of jello, and yet they are held together and held in place by these reticular fibers, so that while they are very squishy and very prone to damage, they can maintain their shape and do their job. Trying to suture a bleeding liver is like trying to sew up jello. It's very challenging to fix damage. You know, it can be done, but it's very challenging. You know, a common injury from blunt force trauma, you know, like being jammed up against some, you know, like the steering wheel or something on the left-hand side of your body, is to rupture the spleen. The spleen is one of those soft, is one of those soft, squishy organs. The spleen's role is to filter out old red cells, recover the iron, recover the hemoglobin. And it is, again, has the consistency of jello. If the spleen ruptures, it tends to bleed out. So conventional wisdom used to say, if we have a ruptured spleen, remove it. Now conventional wisdom is saying, if we have a ruptured spleen, stop the bleeding, treat it, and let it recover, and it will recover. So, okay. This is what loose connective tissue with a lot of reticular fibers looks like. We have that framework, that mesh consists of elastic fibers, reticular fibers, and collagen fibers. That's what helps to hold our abdominal, our viscera in place. Yeah, so we have elastic fibers, we have reticular fibers, and we have collagen fibers in there, all mixed together in loose connective tissue. We would see this loose connective tissue lining the abdominal cavity, protecting our soft, squishy organs, the viscera. The viscera have no protection. You know, the heart and lungs are protected up here by the rib cage, but below the diaphragm, there's nothing to protect any of the other organs. So we depend on this connective tissue to help hold it in place. It is the attaching, if you will, tissue. And we see it in, again, covering the squishy organs like the spleen and the liver to at least let them maintain their shape so they can do their job. It's as little as protection as they get. So now dense connective tissue is made up mostly of collagen. Remember, collagen is the most abundant protein in the body. It is very powerful. It gives us great strength. It works by lining itself up in row after row after row, side by side, looking something like this. If I were going to pick up a 100-pound weight and I had a thread and I tied the thread very carefully to the 100-pound weight, I went to pick it up, we know what would happen. The thread would tear. But what if I had 1,000 threads all tied together, all running up and down like this, and I held on to the 1,000 threads? What are the odds of picking up the weight then? Probably a lot better, because now I have this great strength from all these fibers lined up side by side by side. And this is how the collagen fibers form our tendons. You know, dense connective tissue running parallel in a straight line so that the forces can be applied from the muscles to the bone and move the bone. You know, our muscles alone aren't strong enough to do the things we want them to do. So we have to amplify their strength by using the bones as levers so that we get this great strength out of our muscles. But I'll tell you, these tendons are so strong, they will snap bone before they themselves snap. You know, they will break bones. So, which is, in the long run, it's probably easier to fix a fracture than it is to try and put a tendon back together again. So, Either way, it hurts. But okay, this is collagen. This is what collagen looks like. Collagen is woven together in these straight lines. We find this in our ligaments and our tendons. And you can see in the illustration on the left there, like at the shoulder, we can see where the muscles are attaching to the bone. Now, this is dense regular because we're running in nice parallel lines here. We also have irregular, which is exactly what it sounds like. The collagen fibers are still there, but they may run in multiple directions. Usually we, in our joints, like our shoulder and our hip, we form a capsule around the joint, and that gives our joint tremendous flexibility. You know, consider what our shoulder can do, and we'll talk about this when we get into muscles and bones in lab. But you know, consider that we have this almost 360-degree range you have at your shoulder. Now the socket here on our shoulder is really just like a flat surface, but it's the tendons and ligaments and the capsule that holds everything together, gives us great strength at our shoulder, and still lets us do this wide range of motion. That's what this dense, irregular connective tissue does. It gives you great strength, great range of motion. We also find it underneath the dermis, where our skin, you know, underneath we have the epidermis and the dermis. You know, we have a lot of dense, irregular connective tissue there, because you know our skin has to have flexibility, but also strength. So, okay, here's one that many of us are familiar with: cartilage. Cartilage is a connective tissue that has living cells called chondroblasts or chondrocytes embedded in a gelatinous matrix, sort of like a plastic, if you will. And in our bodies now, we have cartilage at a few places. We have cartilage at the tip of our nose. We have cartilage in our ears. Our airway, our trachea is made of cartilage. Our knees have pads of cartilage between the femur and the tibia. You know, the meniscus, the menisci are pads of cartilage. When we were 6 weeks old, 6 weeks after conception, we were still an embryo, and we were the size of maybe an aspirin tablet, a little tiny tablet about that big. We had a full skeleton in place made of hyaline cartilage. And we used that as the model for laying down our bone for our bony skeleton, that skeleton right there. And from 6 weeks after conception, I mean, we're tiny, but we have a full skeleton in place. 6 weeks after conception, for the next 23 years, we will continue to lay down bone as that skeleton gets bigger, and we won't stop adding to it. We never stop adding to it. But our full skeleton will not be finished until we're 23 years old, more or less. It takes about 23 years to go from the cartilage skeleton, which we laid down in 6 weeks, to the full bone skeleton, full size like we have there. But we still keep some cartilage. Hyaline cartilage is still present at the tip of our nose and our ears and our trachea. We have fibrocartilage, a fibrous or a stronger cartilage between our backbone, between the vertebrae. So we still have cartilage. We have fibrocartilage at our knees, so that it doesn't hurt when we walk and run and do things as the femur sits on top of the tibia. We don't like to have friction. We don't like to have bone on bone. That's not good. So we have these pads of cartilage there. It looks something like this. The cells are called chondrocytes. Anytime you have the word cyte in place as a suffix, cyte means cell. So a chondrocyte is a cartilage cell. Now, chondrocytes came from chondro, chondroblasts. Blast means builder. Chondroblasts lay down the matrix. When we're building cartilage, chondroblasts lay down the matrix, and then they get trapped inside a bubble in the matrix, in their own gel. And the chondroblast turns into a chondrocyte. So what you're seeing here, this is hyaline cartilage, and every one of those little bubbles there has a cell in there. It's a chondrocyte, and the bubble is called a lacunae. So the chondroblast is laying down the matrix and gets stuck. It paints itself into a corner, and which, have you ever painted yourself into a corner? You know? Okay, yeah. Okay, I know. Yeah, it happens. I was painting my garage this summer and said, I better stop or I want to be stuck in a corner for a couple hours. But you know, we paint the chondrocytes chondroblasts paint themselves into the corner. They get stuck and they can't get out, and then they become a functional cartilage cell. Now, the problem with a chondroblast and a chondrocyte is there's no blood supply. They don't have blood vessels. They have to get all their nutrients through diffusion. Oxygen and nutrients and waste products have to diffuse in and out through the matrix, which it does. All of our hyaline cartilage is alive, but it has to diffuse through the matrix, which is why if you have a cartilage injury, it takes longer to heal up, because you don't have the direct, blood going there like to muscles or to bone or something like that. You have to get it through, it has to diffuse through, and it takes time. So cartilage injuries don't heal as well, simply because they trap themselves in these lacunae. Now, bone has a similar arrangement. Bone is a lot stronger, though. The matrix for bone is much, much stronger. And the bone builders are called the osteoblasts. And the living bone cells are called the osteocytes. And they lay down a matrix with calcium and phosphate salts in there. It is very firm. It is very strong. It's almost like concrete. And it wraps itself with collagen fibers to give great strength and flexibility. And it looks something like this. This is what we call compact bone. Now, each one of those black spots that you see there is a lacunae with an osteocyte inside. Compact bone. There's the lacunae, and you have an osteocyte. The difference between this and cartilage is that those little tiny black lines all over the place, those are canals. And so every lacunae, every osteocyte and every lacunae communicates to every other osteocyte, and they send nutrients and oxygen and waste products back and forth through capillaries. So everybody's getting fed, and the nervous system knows what's going on at the same time. So bone, when it's injured, recovers much quicker than cartilage does. Bone is living tissue. We all tend to forget that because we look at that a skeleton. But when bone is alive, it's a living. When we have a living, breathing person, the skeleton is alive. 40% of our skeleton, it consists of organic material like living cells. So, adipose tissue. Adipose tissue gets a lot of bad feelings, bad publicity. Adipose tissue is simply a cell that stores fat. That's all it does. Adipose tissue will enlarge or shrink depending on how much fat it's storing. A person who maintains a high fat or a high carbohydrate diet, the sugars that we eat, if we have excessive amounts of carbs in our diet, the excess sugars from the carbs end up in adipose and it gets stored as fat. But adipose has an important role. It's an insulator. It protects us and cushions us. It keeps us warm in the winter and cool in the summer. It protects us from blunt force trauma. It does a lot of good. It's a great energy source, storage area for us. So it isn't as bad as a lot of people give it, say it is. But like always, too much of a good thing is always never good. You know, if some is good, more is not better. So there's a nucleus, and it has elastic fibers in here. Adipose tissue meets the definition because it has lots of elastic fibers present. Blood. Blood is the last of the connective tissue. It has living cells, it has red cells that carry oxygen, white cells that fight infections. It has platelets that help us to clot when we need to. These are the living cells in the matrix of the plasma, the liquid matrix. The fibers that will form a blood clot for us are in solution. They only get activated when we have a penetrating injury and we form a clot. And so we see here are the three types of blood cells. We have the red cells, the white cells, and the platelets. These little tiny purple dots are the platelets. They're really just cell fragments. They're not complete cells, but they will come together and form a plug at the site of an injury until we can get a clot formed to stop the bleeding. These are white blood cells. We have in every cubic milliliter of blood, we'll have about 5,000 of these white cells. They are the infection fighters. We have white cells that are called natural killer cells. They kill bacteria, they kill viruses, they kill infected cells. And then we have the red cells. These are the red blood cells here. So in that same area where we'd have 5,000 white cells, we'll have about 5 million red cells. Their role is to carry oxygen for us. And they do it very well. Every red cell has red cells generally have a lifespan of about 4 months. White cells can be variable, from a few days to years. These are the three types of cells that we would find inside in our blood. So we meet the definition. Living about 4 months, 120 days. Now, transfused blood only has that's been frozen, only has a lifespan of about a month. But our own whole blood, a red cell will last about 4 months. Apparently, red cells don't like to be frozen and then thawed out and then used. So, okay. The last category of tissues in our bodies are neurons, nervous tissue. They send nerve impulses back and forth. This big thing here is a neuron. Now on the neuron, we have some structures. That's called an axon. The axon is what a neuron uses to send signals. Neurons always send signals away from their cell body using an axon. Axons form what we call the actual nerves. You'll hear all about that when we get into chapter 11 and 12. So, okay. There's the cell body of the neuron. Neurons also don't reproduce. We have all the neurons we're ever going to get at birth. At least we think that's the way it is. That's what conventional wisdom says. We may, in a few years, we may discover that we're actually generating more neurons than we thought we were, but essentially most neurons do not reproduce. If a neuron is damaged or destroyed, it's gone. So there's the cell body. Now these little purple things here are called glial cells. They are the helper cells for the neuron. They circulate cerebrospinal fluid. They fight infection. They provide nutrients for the neuron. They insulate the neuron's axon so that it can send nerve impulses at a faster rate. And there are many more glial cells than there are neurons, because they're all busy working in the central nervous system to help all these cells. In some neurons, if an axon is damaged, the glial cells can actually repair the axon so it can function again. While we say that neurons do not regenerate, some neurons can repair themselves so they can function again. Because if that wasn't the case, how could we ever transplant anything? If we couldn't get the neuron to start working again after we transplant an organ, how can we ever transplant an organ? How can we ever reattach a body part? You know, someone cuts their hand off. How could you put the hand back on if the neurons weren't going to be able to communicate? But in some neurons, we can, even if the axon's damaged, we can put it back together again, and the neuron will take over the job of repairing it, and it'll work. So, okay, let's see now. There's the nucleus, and these what we call nerve fibers, are the receiving end of information. They're called dendrites. When a neuron communicates, it receives information from another neuron on its nerve fibers, and then sends that down through the axon to talk to another neuron, or to a muscle cell, or to a gland. So, and generally, most neurons all look about the same. Okay, this is our small intestine again, a little a better close-up of this. And you can see when I said that there were 2 layers of muscles in the small intestine, you can see them here. This is the outer covering of the small intestine out here. That's got that serous membrane I think I mentioned in lecture when we wrapped up chapter 1. Right there's the serosa or serous layer. Again, it's a lubricated layer, but underneath that, we have a muscular layer, and we have that arrow should actually be over here a little bit. We have the longitudinal layer that runs lengthwise, and we have the smooth muscle layer, the circular layer that runs around. And there's the circular layer right there. And so this will run through the same pattern applies all the way through our digestive system from the esophagus down to the anus. The only difference in our digestive system is at the stomach, where we have a 3rd layer that is sort of oblique. So we have a 3rd layer of muscle. And then once we move through the muscle layer, now we're going to be exposed to the outside again. We start moving into the area of epithelial cells, where we run into, instead of serous membrane, we run into mucous membrane. And if you look carefully down here, these are all simple columnar cells here, and these little light structures here, stained light pink, I guess. These are all goblet cells in here all the way around. So we use those goblet cells with the columnar cells a lot. So, and of course, we call that the mucus layer because of all the mucus from the goblet cell. Here is the lumen of the small intestine. Now, just a few comments here on the skin. I don't want to go into this. I want to get that stuff weighed. Skin, we have 3 layers in the skin. We have the epidermis, the hypodermis. We have the epidermis, the dermis, and the hypodermis. The epidermis is essentially the dead layer. Most of the epidermis is dead till you get down to the basement layer of the epidermis. We start having living tissue in the dermal layer, and we have our adipose in the hypodermis layers. In this model here, or this illustration here, we can see there's our dermis. Nothing is living until we get right down to the edge here, this up and down edge. You'll notice also that there are blood vessels are finally showing up here. Blood vessels don't occur until we get into the dermis. So the area between the dermis and the hypodermis is a very fine line where we have living cells pushing up the dead cells on top, and then they keep sliding off. There we have the dermis. We see where we have hair follicles, we have sweat glands, we have oil glands, we have blood vessels. When you cut yourself and you're bleeding, the only way that you bleed is if the injury penetrated the dermis. Now, we're not talking deep layers of skin here. But we are bleeding. If we're bleeding, we penetrated to the dermis. If we're not bleeding, then we didn't know we were injured. You know, we didn't get down that far, you know. A paper cut always hurts, but a paper cut may not reach to the dermis, and then we may not have any blood. We may not even feel it if, you know, I mean, think of all the times that we would injure ourselves and we're not aware of it because we didn't get down to the receptors or to the blood vessels. Because something else about the dermis, all the touch and pressure and pain receptors are generally located in there. And then there we have the hypodermis, where we have the end of the hair shaft, and we have lots of adipose tissue there for storage and insulation and protection. Okay, now, those are one of our touch receptors right there. There's a hair follicle. Now notice the hair follicle. The hair follicle has the hair shaft in there. Lots of people will say that are concerned about their hair coloring, and an individual that as they get older will say they have gray hair. Well, there's no such thing as gray hair. Gray hair is a hair shaft. When it loses its pigmentation, doesn't go gray, it goes transparent. And it appears gray because of light rays bouncing off through the hair shaft and all the other hairs and maybe off the scalp. So it looks gray. Someone who has lost all their pigmentation and still has a full head of hair, their hair appears white. Well, it's the individual hair shafts are actually completely transparent. There's no such thing as white hair. It appears white, but it's actually transparent. So, and so what will happen is as a person ages and they start losing their pigmentation in their hair follicles, then their hair starts like, oh, you're turning gray, you know, on top or on the sides or whatever. It's a function of life, you know. Some people don't have an issue. Some people have an issue very early on in life. My dad had, he had a shock of gray hair that started right here and worked its way back before he finally turned fully gray. It took him years to get that. It's a genetic thing. A lot of my family on my dad's side had that same issue, including one of my female cousins had that. You know, it started when she was 19. So she wasn't happy, but it happens. You know, lots of things like that happen. I had a friend in college who started developing his male pattern baldness in his sophomore year. And by the time he was a senior, I mean, it was like, you know, gone. Yeah. Nobody wants to lose their hair at 19, but it happened. You know, nobody wants to have gray hair at 19 either, but it happened. Anyway, hair shaft. This other thing we see here in the hair shaft is an oil gland. That's what makes our hair oily. We don't wash our hair frequently. Our hair gets oily and dirty because the oil, which is put out to lubricate our hair and make it flexible, also makes it slippery and dirty because the dust and dirt and debris are picked up by the oil, and you know we have to clean it. Otherwise, when you go to bed at night, your head hits the pillow, slides right off because it's so greasy. We don't like oily hair, even though the oil is there for a purpose to protect us. So we also have a adipose, I mentioned that. Okay, here we see, okay. These are the various layers of the epidermis. The stratum basal layer is the layer right above the dermis, the epidermis. Stratum basal for basement layer, that's where we have living cells. Directly below that is where the dermis starts. So we have living cells in this very bottom edge here of, you know, this up and down area. This little purple band here is the living area. So, okay, now, okay, here we have a sweat gland, an eccrine sweat gland. That sweat gland is designed to release sweat so that we can cool down when we get too hot. You know, it's a temperature regulator. We have lots of sweat glands throughout our bodies so that we can try to, you know, to help regulate our temperature when we get too hot, and we stop sweating when we get too cold. And there's, you know, We release it through pores. Something else we see, now this is what's known as an erector pili muscle. This muscle is what causes goosebumps. Our hair stands up on end when we get cold, sometimes when we get scared. But our hair stands up, the erector pili muscle contracts to raise our hair shaft to trap air on the surface of our skin. Air is an insulator. And by doing so, we can trap a layer of warmer air around our skin. It doesn't do a whole lot, but it does do a little. So that's why when we get cold, our hair stands up, when we get these bumps on our skin. So thanks to these muscles, these erector pili muscles. Okay, next oil glands again, and the hair follicle again. And the root. Okay, one thing I want to show you on this model, I have to go back a few slides to get to it. There we go. There we are. Now, there are a series of bumps right here. And they are called the dermal papillae. These bumps are what form the loops and the ridges and the whorls of our fingerprints. The dermal papillae are at the layer of the dermis and epidermis where they join together. And the bumps on the dermal papillae are going to form the shapes that we have underneath our dermis that forms our fingerprint layer in here that doesn't go away. You know, our fingerprint is genetically determined. It's an identifier for us. And for years, of course, fingerprints were used before DNA testing, but of course, fingerprinting is a whole lot less expensive than DNA testing. So most, fingerprints are unique to each and every one of us. And we still use fingerprinting for identification. So, but those are called the dermal papillae. So now let's, and that, we already mentioned all that. Okay, now before we do anything else, let me weigh these two, and then looks like we're going to be doing cheek cells next week. I can talk faster and cut stuff out today, and I'm still 20 after. So, okay, this is this one is the distilled water in the 10% salt solution. And so water should have left the cell. And I'm going to weigh it. The distilled water cell weighed 30.2 grams initially. And it dropped to 29.8. Not much of a drop. .8. So it dropped about a half a gram. Let's measure the salt water one. The saltwater cell weighed 33.8 grams. And now it weighs 37 grams. So that one worked very nicely. We went from 33.8 to 37. So it was about I can't even do the math right. Yeah, about a 6 gram increase in water. So that one worked very nicely. We saw water went into the cell with the salt water inside of it. Because there was lower water inside the cell. Lower water inside the cell and higher water outside the cell. Water came in. We saw a 6 gram weight gain, or 5 gram weight gain. So it worked. So the other one, not so much. So, okay, that's all I've got. Remember, there is no class on Monday. Monday is Labor Day. It's a holiday. No one will be here. So, okay. And I'm going to get us out of here now.